When we first learn about genetics, we're taught a highly predictable set of rules. You inherit traits from your parents, and those traits resolve themselves in a strict hierarchy, where the dominant versions simply overpower the recessive ones. This framework comes directly from Gregor Mendel. His law of dominance states that when an organism inherits a pair of dissimilar factors for a trait, one member of the pair always dominates the other. The dominant trait expresses itself in the offspring, while the recessive trait is completely hidden. Mendel paired this with the law of segregation. This rule dictates that during reproduction, these paired factors separate entirely cleanly. An offspring receives one distinct allele from each parent, with absolutely no blending of the two original traits. Because this model works so well for basic inheritance, we fall into a cognitive trap. We start to imagine alleles as fixed entities, permanently stamped with dominant or recessive labels that dictate exactly how they will behave in a cell. But treating dominance as an intrinsic, autonomous property of a piece of DNA severely limits our understanding of biology. To see why, we have to look past the macroscopic traits and zoom down into the molecular reality of the cell. At this scale, a gene is simply a sequence of information. Its primary job is to produce a specific physical product, usually a protein or an enzyme. Dominance is an illusion of scale. It is not an active battle between two competing genes, but a retroactive description of how well those resulting enzymes process their chemical targets. To understand why Mendelian rules frequently break down, we have to track the biochemical output of the alleles, rather than just looking at the final physical trait. Humans and most complex organisms are diploid, meaning we carry two copies of every gene. In a heterozygote, those two copies are not perfectly identical. This diagram shows the baseline function of a gene. The normal, unmodified allele produces a functional enzyme. That enzyme perfectly matches its target, a specific chemical substrate, and successfully alters it to create a biological effect. The second copy of the gene, the modified allele, has undergone a mutation. Instead of a functional protein, it produces a deformed enzyme that can't attach to the substrate, or perhaps it produces no enzyme at all. Even though one allele is broken, the single working allele produces enough functioning enzyme to process the substrate and hit the required physiological threshold. The physical trait appears completely normal. The broken allele isn't actively suppressed by the working one. The working allele simply produces enough biological material to get the job done. This mechanism of biological sufficiency is what we macroscopically label as complete dominance. We can observe what happens when an allele can't produce enough material by looking at flower color in the snapdragon plant. This diagram maps a classic experimental cross. When a true breeding red snapdragon carrying two capital R alleles reproduces with a true breeding white snapdragon carrying two lowercase r alleles, it yields an F1 generation with a mixed RR genotype. Every single offspring grows pink flowers. Using our enzyme model, the cause of the pink color becomes clear. The single red allele only produces half the required enzyme. It is biologically insufficient to fully saturate the petals with pigment, leaving us with a diluted intermediate color. If we let these pink flowers self-pollinate, the F2 generation emerges in a strict mathematical ratio: 1 red, 2 pink, and 1 white flower. This 1:2:1 phenotypic ratio completely breaks Mendel's expected 3:1 outcome. The genetic material itself never blended. The individual R and R factors segregated cleanly into the offspring, just as Mendel predicted. The macroscopic expression simply fell short because of an enzyme deficit, a process called incomplete dominance. Sometimes an offspring perfectly resembles both parents simultaneously. We see this in human biology when typing blood. The human ABO blood group system is governed by a single gene, but across the global population, there are 3 distinct variations, or alleles, for this gene: IA, IB, and a recessive lowercase I. This 3D cross-section shows the surface of a red blood cell. The IA and IB alleles act as active builders, each synthesizing a slightly different type of sugar polymer that protrudes from the plasma membrane. The recessive I allele builds nothing at all. If you inherit both the IA and IB alleles, neither enzyme interferes with the other. Both alleles independently build their specific sugars and attach them to the cell membrane side by side. Because both enzymes are perfectly functional, the cell expresses both outputs simultaneously. This codominance proves that alleles are not locked in a battle for control. If they work, they simply execute their programmed functions independently. We can push this biochemical model even further by examining pleiotropy, a scenario where one single gene product drives multiple distinct effects. A clear example is the synthesis of starch in pea seeds, which is controlled by alleles B and B. This split-screen diagram allows us to observe the exact same heterozygous pea seed, containing one B allele and one B allele, from two completely different physical perspectives. On the left, we observe the macroscopic shape of the seed. The capital B allele synthesizes starch very effectively, while the lowercase b is inefficient. Yet the single B allele produces enough starch to keep the seed plump and perfectly round. At this scale, the B allele exhibits complete dominance. On the right, we zoom down to the microscopic level to measure the actual size of the individual starch grains inside that exact same round seed. Because of the inefficient B allele, the total starch output is reduced. The resulting grains are intermediate in size. At this specific scale, the relationship is one of incomplete dominance. This single heterozygous seed presents a direct paradox. The exact same alleles are completely dominant when determining the shape of the seed, but incompletely dominant when sizing the starch grains within it. The gene itself didn't change its behavior. The DNA sequence and the enzyme it produced remained entirely static. The only variable that shifted was the specific biological metric we chose to measure. Mendel's initial laws gave us a reliable framework to track traits through generations. But as we move from the diluted pigment of a snapdragon to the co-expressing sugars of human blood, down to the dual nature of a pea seed, a larger reality emerges. The terms dominant and recessive are highly useful macroscopic shorthand, but they are just that, shorthand. They do not describe the physical mechanics of the genome. Dominance is not an autonomous feature of a gene. It is a strictly context-dependent relationship, defined entirely by an enzyme's efficiency and the specific trait we decide to observe.